Hello, everyone. My name is Dan, and I'm happy to share with you guys reading in my final MoCS project, which is entitled Multi-Species Epidemiological Models to Investigate the Dilution and Reservoir Effects in Amphibian Disease. Although our model isn't just applicable to amphibian diseases, including the one I'm about to talk about, but could be used for any disease that affects multiple species, where the species themselves are affected differently by the disease. So the specific problem that we were looking to study is the disease chytridomycosis, and we believe our model is very applicable for studying this disease and others. So chytridomycosis is a fungal disease that affects a number of amphibian species, leading to massive die-offs and loss of biodiversity in habitats all around the world. And the key about chytridomycosis' spread, which is one reason why our model is particularly applicable to study it, is the way that it actually spreads. So unlike most epidemic spread that we model with the classic SIR model, for example, where the rate of infection is influenced by interactions between susceptible and infectious individuals, chytridomycosis is spread by these motile zoospores that are present in the water and could stand to affect species all over the particular population, and all different species as well. So that's one reason that we thought this model was exceptionally applicable. And part of the complexity that it plays into it is that species all around the world are differently affected by chytridomycosis. So here we see the magnitude of decline across all different species, and we see that species are affected very differently all around the world, evidenced by this difference in the severity of the decline. So this brings us to the question that we wanted to study, and here I'll just be discussing some of the models that we use to kind of establish these principles, those of dilution and the reservoir effect. But our question is how the species composition at a site influences the epidemic's spread of disease. And we are also interested to see if there are migration in between neighboring sites, how different species can spread the disease, not just in one individual sort of compartmental models, but those with neighboring sites. And our eventual goal is to make this into a network model, although that didn't materialize in time for this presentation. So I kind of want to go into what this dilution effect is, since this is a relatively new concept in disease ecology, and one that I just became familiar with here for this project. But the dilution effect makes predictions about how the species richness at a site influences the spread. So here there's two examples, that of managed dilution effect. By introducing more biodiversity into a site, you could actually decrease the transmission of disease, whereas the natural dilution effect can lead to which occurs when diversity is inadvertently lost. You could actually increase transmission because you are getting rid of potential diluters, right? Species that don't actually spread the disease particularly well. So this brings us into the actual numerics of our model of our model itself. So in the examples I'll show here, we have a classic SIR model where instead of the rate of infection being influenced by the number of susceptible and infectious individuals, it's related to this value called community competency. So each species has a maximal infection rate that we'll call beta here. And there's the number of susceptible individuals and also this community competency, which we're saying is related directly to the proportion of infectious individuals in the population. So the key assumption that we make for this model is that the populations are initially at a steady state, so that this proportion of infectious individuals could give us essentially an idea of what the magnitude of zoospores are, since in a given pond or something of a given size with a total abundance of amphibians, right, the proportion of them that are infectious is probably related to, is rather like directly related, is what we're assuming, to the number of infectious individuals. So that is what indeed determines the spread. So additionally, each species has a intrinsic recovery rate. So you can imagine that species with a lower gamma stay in the infectious compartment for longer, thus producing more zoospores and affecting the disease spread for other individuals in the population. So here we see the effect of the initial proportion of infected individuals. So since the rate of spread is directly related to this proportion, if you have more infectious individuals to start, the rate of disease spread is faster. Here we are changing the gamma term, so that's the rate of recovery. And we see when there's only a single population or a single species in the population, the species A, although the rate at which or the total number of infectious individual declines more quickly when gamma is larger. The epidemic spread evidenced by the number of removed individuals isn't particularly different in between the different gamma groups, and in all three of these cases, the populations go to basically extinction within 100 time points. So here is where we bring in multiple populations into the model. So we have our initial susceptible population, and here we have another population, one that's very similar to our initial population, and another where this gamma is lower. And here, when we have multiple populations, we see that there is a relationship in between the gamma, right, the removal rate for this introduced population, influencing the rate of disease spread for both populations. So we could call in the group on the right, this species B is acting as a reservoir. And here we see that the initially infected population has an influence on the rate of disease spread. So if we say have our initial susceptible population infected first, there is a relatively lower rate of disease spread than when the reservoir population is infected first. So when introducing populations into when different species are introduced into a given ecosystem, either through invasion into the area, and if they are good reservoirs for this disease, we see that has an influence in our model here. And here we're going to be modeling the dilution effect by including a unaffected diluter species, and by changing the size, right, essentially our dilution factor, by introducing these less or unaffected species that influences the disease spread for the susceptible population. So in this case, the more unaffected or lesser affected individuals affects whether or not this disease reaches epidemic proportions for our susceptible species. Here is a look at our 3-species model, where we have both a reservoir, or all three, a reservoir, susceptible, and diluter species. And depending on the species that is initially infected, the one that's first carrying the disease at an affected site, that influences the spread of the disease, although less so than when we only had 2 populations. So in reality, we probably see many more susceptible species in one place, and there's probably more complicated interactions in between species.